Thank you all for being here. The College of Science Office of Diversity and Inclusion is really excited to host Dr. Kory Evans, who is an assistant professor at Rice University. And he's an evolutionary biologist that's broadly interested in the development, evolution, and ecology of phenotypic diversity in teleost fishes. His research integrates developmental biology, biomechanics, phylogenetic comparative methods, and ecology to understand how phenotypes develop and evolve and interact with their respective environments across multiple time scales, and how intrinsic development and extrinsic environment mechanisms influence patterns of phenotypic diversity. So today he's going to be speaking to us about his research on the evolution of the fish skull and his journey through academia as a Black scientist. Then after his talk, we're going to have a Q&A/informal discussion where you guys can ask any questions you have, and we have a few questions that we also want to ask Kory. So without further ado, I'm going to hand it over to Kory.
Hey everybody, my name is Kory Evans. I am a professor at Rice University, and today I will be walking you through kind of my journey through academia and my journey through research in general through the lens of the fish skull and fish skull-shaped evolution. So let's start with my origin story. Everybody needs an origin story. So I was originally born and raised in North Philadelphia. It's kind of your typical inner city environment, concrete jungle. For those of you who are familiar with the Philadelphia area, I grew up one block away from Alny High School. And it was Philadelphia is a really interesting place because unlike larger cities, the kind of city planning of Philadelphia actually included a couple more green spaces, or a little more green space than a typical city. And as you'll see in the next few slides, that actually really came in handy. So in my household, competition and sports reigned supreme. My parents believed that kids should be outside all the time. So they had us doing all the sports: soccer, basketball, martial arts. Sometimes in martial arts, I'd have to fight my own siblings. So it was like there was four men in the household, and it was ridiculously competitive. Everybody did a sport. My mom did competitive race walking. If you were swimming in the pool, in like a competition pool, sometimes you might be racing mom, dad, your brothers. You never knew who, but you were competing with somebody. And that kind of that competition and that sports was kind of really woven into the fabric of my household. What was really cool though is kind of was this reverence for nature that was really held at I would argue equal importance in our house. So because my parents believed kids should be outdoors, my parents are really big into taking us into outdoor spaces. And Philadelphia had a lot of green spaces to take us to, relative to other cities. So my parents would take us to creeks, and they'd still have us, practicing. So if we had track practice, we'd run along Kelly Drive, which is a really nice kind of scenic jog. And yeah, so my parents were always really big into taking time to kind of just be in nature. Furthermore, during my, treks and journeys through these natural spaces, I kind of picked up this love of fishes. My favorite places to practice and jog and run and train were places where there was water running, small streams. And as I had moved into high school, my grades were really bad. My grades were really bad at first. And one of the few things that I was allowed to do was go out and take walks or go to pet shops. And that's kind of where my love of fish keeping really took off. In fact, pet stores are really kind of like my one little kind of getaway from constantly being on punishment for having bad grades. My parents were, we had cats in the house, so we had to get the cat food. I was like, oh, I'll get the cat food. I'll just run up to PetSmart. And my mom would ask, why is it taking you 2 hours to go to the pet shop? Because I would just stand there and stare at Oscars all day. And eventually, I got my own little summer job, and I started bringing home fish, and setting them up in my aquarium. So fast forward to about 2007, 2008, I decided, maybe I really like fish. First of all, I was like, I think I really like fish. Maybe I should take this show on the road. So I found this. I went to a military academy for high school, and they were constantly trying to recruit us with these National Guard pamphlets. And I remember one of these pamphlets at the back had this very obscure small university, and one of the majors that it was being advertised as marine biology. I had no idea where the school was, but this is like the first place I saw that had a marine biology program. After a quick Google session, I found out it was in South Beach. I was like, yeah, let's go. So it was actually in Fort Lauderdale, Florida. I realized that Miami was a far larger place than I had imagined as a high schooler. nonetheless, I took my talents to South Beach in about 2009. I went to Nova Southeastern University, studied marine biology, and that was really a phenomenal program because it was about 20 minutes from the beach. So all of our biology, like labs and field trips, like half of them were always at the beach. We were snorkeling, we were fishing, we were staining. It was awesome. Even invertebrate zoology, which had no vertebrates whatsoever, was still a very exciting class. Uh, so, uh, so I did 4 years there, and I finished in 2013. Then I moved west, and again at the time, again coming from North Philadelphia, anytime I moved west really was usually like the furthest I'd ever been west. So when I moved to Lafayette, that was the furthest west I'd ever set foot, and certainly the furthest west I'd ever lived. So I did a PhD in University of Louisiana Lafayette starting in 2013, and that's where I met my advisor James Albert, who was one of these really big, deep evolutionary biology thinkers. And that was kind of my first time in academia realizing that, different professors and different people and different departments have different ways of looking and thinking about the same process, which is evolution and ecology, and how organisms kind of track their environment, how they evolve through time. So a lot of my upbringing in undergrad was really geared towards ecology. We had the beach right there, so we have this crazy coral reef ecology program, and a lot of our classes were kind of sparse on evolution. But when I met James, James is really all about evolution. Man, all he cared about was at the time was where species were on the map, and how they evolved through time and who was related to who. And that was a really kind of stark contrast because when I got to that lab, I wanted to do ecology. And James was like, well, all these fish live in murky Amazonian waters, so you're not going to be able to like scuba dive with them. I was like, I see. Nonetheless, hanging out in that lab really got me thinking deeply about evolution and how traits evolve. It also helped that the fish that James specialized in, that the Albert Lab specialized in general, were very, very weird fish. So James and the Albert Lab in general, they specialize on this clade of fishes called the gymnotiformes. These are your neotropical electric fishes. So these fishes are capable of generating weak electric currents, and we'll get into that in a bit. But so I'm walking into this lab for the very first time. There's these weird fishes in jars with these crazy faces and crazy body shapes. And so for the first like 2 months in lab, I was just overwhelmed all the time. I didn't know anything. I spent 4 years learning all the fish that lived in coastal Florida, and I had no idea what any of these things were. All their Latin names are weird, and I had no idea. I had never been to where they're from, so I couldn't even picture them swimming in their environment. And I couldn't picture anything swimming with a fin like that in the first place, right? And let alone we get to the faces, and these faces are all bizarre. So everything about these animals was weird. And James, I remember, I think my first month in lab, James had actually given us a really formative assignment where he wanted us to sort a bunch of fishes out of these buckets from an old field expedition into lots based on overall similarity until we could actually ID the species. And that actually ended up being a great activity because I had my hands on a ton of fish. So I'd pick them up and I look at their faces. I was like, whoa, man, what are you eating? What are you using this face for? Again and again and again. And it took like 2 weeks to sort all the fish or something like that. And I started really kind of forming these questions about how these different phenotypes were evolving. And for me, I really zeroed in on the head. I don't know why, but I'm just really into fish faces. That's really about it for me. And so I started asking these questions, and a couple questions kept jumping out again and again and again. Some of these questions were, how did they get these faces in the first place, and what are they using them for? And these two questions really ended up being, I would argue, kind of the basis for the rest of my research, even to this day. So after I kind of had those questions, I decided to start working towards answering them in a dissertation format. So the roadmap for the majority of this talk will really be centered on those two questions. First question being, how did the electric fish skull evolve in the first place? We know that it's weird. I showed you that it's weird. And that was one of the things that I was really kind of enraptured about. The second was, how does the skull interact with its external environment? And the simpler form of this question really is, what is it using its skull for? What are electric fishes using their skulls for in their natural environments? And these two questions were very broad and very vague, and therefore resulted in a lot of questions, more specific questions that spun off of them. So let's talk about these electric fishes again for a sec. So electric fishes are a clade of weakly electric freshwater fishes. We have other weakly electric fishes over in Africa. This is the better clade. They have cooler faces, And if you notice, and by the way, these fishes are distributed in Central and South America. So if you notice, what's one thing that's really striking about these electric fishes, other than the fact that they can generate electricity, is their crazy craniofacial diversity. So here you can see I have arranged in phylogenetic order multiple independent evolutionary instances of long and short faces. And this is a prevailing theme across Junoniforms, and it will be a prevailing theme across this talk for sure. So to answer the first question, I set out all these fishes, and I started thinking, how did the electric fish skull evolve, and more specifically, how do patterns of craniofacial variation scale to diversity? Because you see, variation is actually the raw material that evolution acts upon. So how do you go from intraspecific variation to interspecific diversity? That was one of the questions I was really into. So what exactly is variation? So when I use variation in this talk, variation gets used a lot. It's a very common word, but in this talk, I use variation to refer to any difference within a species, so intraspecific. So variation is any difference between individuals, basically. And domestic dogs, I use this example a lot. They're really a poster child for variation because they come in all these different shapes and sizes, and they're all of the same species. Now, domestic dogs were artificially selected to be this way, but they're still really, really variable. So if you're thinking about variation, if you're thinking about the kind of contributors to the variation that an organism might experience over its lifespan, one of the largest contributing factors to an organism's total variation that it experiences is ontogenetic variation. So the shape change or the change that an organism experiences through growth can oftentimes be very, very dramatic, right? So think back to yourself, or if you have children, or if you have siblings, you might notice that a couple years ago, those small babies look a lot different than they do now, and that's because a lot of organisms really do experience a lot of difference or a lot of change in growth. So here I have two electric fishes that I captured in Panama. I caught them from the same floating root mass at the exact same time, and they actually look quite a bit different. The specimen on the top has a very rounded skull, and the specimen on the bottom has a very elongated skull. So if you were in a museum collection rummaging through jars and these animals somehow ended up in a different lot, you might not immediately recognize them as being part of the same species. However, one is simply a juvenile of the other. And as you can see, this juvenile looks dramatically different from this adult. So this was really just an example to show you how dramatic ontogenic variation can be within species. Another cool thing about ontogenic variation is that the magnitude of variation can differ between species. So some species change a lot in development and others don't. And this can have some really interesting implications downstream for evolution. So changing in development and variation in development is all well and good, but how do you translate ontogenic variation to evolutionary diversity or interspecific diversity? Well, one way you can do that is via developmental bias. So a developmental bias favors the production of one phenotype over the other, such that the developmental programming is capable of producing one phenotype over and over and over again via different means. So when you find a developmental bias, what you often find are multiple what are called developmental trajectories. They're capable of producing the same phenotype over and over again, but they get there different ways. So because developmental biases can produce similar phenotypes, they can actually result in a higher frequency of convergent evolution towards a particular phenotype. And because, and we'll kind of unpack that term convergent evolution in the next slide, but because the developmental programming is driving the convergent evolution, these phenotypes can actually be widely adaptable because it's not external environmental stimuli that are driving this convergence. So what is convergent evolution? I just used that term. Convergent evolution refers to the independent evolution of a similar trait or function. So very broadly speaking, in a functional context, flight in vertebrates is an instance of convergence. Very frequently, when you see convergent phenotypes, it's because they're usually tracking a very similar environmental stimuli. So things that live in similar habitats might look the same, but this isn't always the case. As I just mentioned, developmental biases can sometimes give us convergent phenotypes as well. Electric fishes, Junoniformes, are no strangers to convergent evolution. They do it, they live it, that is their whole life. So here I have just kind of a diversity photo of multiple independent evolutionary instances of convergence to long-faced or long-snouted phenotypes, which I refer to here as dolichocephalid. So here, most of these species are not each other's closest relatives. They're distantly related. I think there's only one case where they're the same genus, but juniforms have evolved long faces again and again and again. It seems to be a very recurrent theme in their evolutionary history. I wasn't the first person to notice this. The long-snouted phenotype was the first thing that jumped out at me when I started in lab because these fish look so weird. But I wasn't the first person that noticed it was convergent. Max Ellis had actually noted this in one of the very early phylogenies for electric fishes back in 1913, and it's since been expanded upon to include the long-snouted mormyrids over in Africa, which are not particularly closely related to gymnotiforms. However, when I was in lab, I was actually working on a taxonomic project, kind of off to the side. And one thing that I was beginning to notice is that there was a ton of rampant taxonomic confusion amongst all these short-snouted fishes. So I was thinking, well, we know that long-snouted fishes are convergent. They're mainstream. Everybody knows that's where the action is. But there looks like there seems to be equal, if not more, taxonomic confusion involving these short-faced species. So it got me asking, well, what about brachycephaly? So in order to answer this question, I put together a crack team of scientists that usually they were all local, including Persanta, who now has a beard. And I asked this question, why the short face? Why are we seeing convergence, or why are we seeing so many short faces in the gymnotiform evolutionary history? So in order to answer this question, I decided to break my project and break this paper into two different parts. So the first part I was really interested in was the phylogenetic aspect of convergent evolution. I wanted to quantify skull shape at the macroevolutionary level, and I wanted to quantify convergent evolution at the macroevolutionary level. So the way that I did that was I used two-dimensional geometric morphometrics at the time. Back then, it was all the rage. But as you see, as we'll progress through this talk, the methods have certainly evolved. So at the time, I only looked at the neurocranium, which is basically the upper jaw, even excluding the premaxilla. The neurocranium was my entire world, and those like 20 landmarks, that was like my whole life for the next 4 years in grad school. So I looked at 133 species, and I analyzed their skull shapes using radiographs and cleared and stained images. And then I took a smaller sample of 17 species, many of which included the long faces and the short faces, so all the convergent phenotypes, and I looked at the development, so the ontogenetic shape change in development of some of those convergent phenotypes. The question that I wanted to ask was, do these convergent adult phenotypes all develop the same way as well? So first I wanted to look at kind of the broad scale patterns of shape diversity and convergence, and then I wanted to look at the underlying developments that form these phenotypes. So the next thing I was really interested in was these kind of concepts of integration and modularity that I had kind of really just stumbled into because I was scrolling through a list of functions using Morpho-J. So I was really interested in how different parts of the skull were covariating throughout development. So when we're studying patterns of trachovariation and patterns of trachovariation in development and evolution, they can really be broken apart. These two patterns can really be broken apart into two different classes, if you will. So the first pattern of covariation we'll be talking about is integration. And we'll be circling back to this towards the end of the talk as well. So integration, especially in ontogeny, refers to the pattern where there's a high degree of covariation between trait complexes and between structures. So changes that occur here in module 1 or trait 1 are expected to be reflected over here in trait 2 because there's a high degree of covariation, usually due to pleiotropy, that connects these two trait complexes. The next case that we'll be talking about is modularity. So modularity used to be kind of referred to as the opposite of integration, but I like to think of it more as a special case of integration. So modularity occurs, there's a high degree of covariation within a particular trait or trait complex, such as, say, if a mutation happened here, it would be fairly evenly distributed across all of its components, whereas there's a low degree of covariation between traits. And this actually has some very interesting implications at the evolutionary level. Right now at the developmental level, what you see when things are modular is some traits might be changing and others might not be. But we'll keep it there for now. So in the interest of quantifying integration modularity, I had to break the skull down into different parts. So me and James came up with a very simple model for skull shape modularity for the electric fish, and we divided the skull into two modules, the face and the brain case, and we tested, and we did find a good signal of modularity between the two. So this was a good hypothesis. Then we quantified developmental covariation using a couple different metrics that I won't get into here, but if you have any questions, please ask me at the end of the talk. The next thing we wanted to do was quantify convergent evolution in some kind of quantitative framework. A lot of times, especially earlier when we would see convergent phenotypes, you would just say that has a long face and that has a long face, and they're not each other's closest relative, therefore it's convergent. And I wasn't really happy with that. I wanted to figure out a better way to quantify shape and to quantify convergence. So we fit some axes of our shape data to an OU model, which allowed us to model convergent evolution. Next thing we used was a phylogeny. If you're trying to study convergent evolution and you don't have a phylogeny, you're going to have a bad time. So we used the highest resolution phylogeny that was available to us at the time, and it was really species dense and very helpful. So let's get into some of these results for our first question. So when we analyzed our phylogenetic sample of skulls across all of our electric fishes, all 133 species, we found that the primary axis of shape variation, or the most important axis of shape variation, was variation between long and short skulls. So the next thing we did was we actually took that axis, that PC1 axis, and just plotted it across the tree. So here you can see a phylogeny, a color-coded phylogeny of skull shape evolution for genodiforms. So the red colors correspond to short faces, and the blue correspond to long faces, basically. And green are intermediate values. So what you can see right off the bat is that both reds and blues appear multiple times independently across this tree. So now we know for sure that there is definitely convergent evolution along this axis of long and short faces in genodiforms. So that was a really nice, that was like the first kind of major takeaway from this analysis. So we decided to kind of look at this a couple different ways. So here we have our same PC1 trait data where the lower values correspond to short faces and the higher values correspond to long faces. We just wanted to look at kind of the magnitude of transitions to long faces versus short faces. So as you can see, there's still rampant convergence. It's the same data, but using this kind of image, you can see that there appear to be way more independent evolutionary transitions towards these short values or these lower values of these short faces than there are to these long faces. But that was fairly qualitative at the time, so I wanted to figure out a more quantitative way to assess that. So we analyzed our shape axis using this program called Lambda1OU, and we produced this very, very busy figure. So I'm going to break it down for you. So basically, all the histograms to the left of this line here correspond to brachycephalic or short faces, and all the histograms to the right of this line correspond to long faces. So the second thing that I want to draw your attention to are the color of these different histograms, which correspond to something called a convergent regime. So convergent regimes occur when two species have similar trait values and are not particularly closely related. They get assigned to a single convergent regime, and these convergent regimes are delimited for us by color. So the first thing that we see here is that there are way more convergent regimes in the brachycephalic category than there are in long faces. So there's one particular regime, this light blue, that appears in nearly every major clade genodiforms. So every major clade genodiforms has evolved a short face, and most of them have evolved a very similarly convergent short face. So that's really cool. And when we look at the convergence towards elongated faces, we see far fewer instances of convergence. So the next question I was asking was, how do these convergent phenotypes develop? So that involved looking at the change in shape over size. So here I just have an ontogenic trajectory. I'm going to break this apart into fewer species, but I just wanted to orient you to what an ontogenic trajectory looks like in case you're not familiar. So here we have size on our x-axis and we have shape on our y-axis, and this is literally just how the organism changes as it grows, how the shape of the skull changes as it grows. And in this case, we're particularly interested in the slopes of these trajectories. So first we looked at the dolichocephalic, or the long-skulled species, and we were really, again, interested in these slopes. And the first thing that we found was, for the most part, across all these different genera and families, if you're growing an elongated skull, you're typically growing it in the same way, right? You have a very similar slope. There's only one slope that differs here. But for the most part, if you're growing a long face, you do it in a very, very similar way. So there's really one or maybe two ways to grow a long face in juveniforms based on our data. However, when we look at for shortened faces or brachycephalic species, what we see is a very, very different story. Here, there are at least 6 ways to grow a short face, which was again really, really striking. And it showed that at least in our data, there are far more ways to develop a short face than there are a long face. So when we started looking at ontogenetic integration and modularity, the first thing that we found was that the degree of integration in your development significantly predicted your adult phenotype. So if you had a long face, turns out you were more integrated in ontogeny between your face and brain case. And if you had a short face, you were more disintegrated, or in some cases, modular. So when we reconstructed some values of integration modularity across the tree for our 13 species, we found that different aspects of their we found that different species evolved different patterns of ontogenetic integration independently, nearly mirroring our convergent evolution. So what is going on, right? So why are these long faces more integrated, and why are short faces more disintegrated? So it got me thinking, how are we getting these, especially short faces, because it seems as though we're getting a lot of action happening in short face land. So how are we getting all these short faces, and how are we getting all these short fish faces? So I turned to the developmental literature to answer this question, and I found this really interesting mechanism where sonic hedgehog signaling from the forebrain it leaves the forebrain and hits this FEZ, which is just a kind of a facial primordia. And when the sonic hedgehog signaling reaches the facial primordia, it prompts the face to expand, and you get a regular chick beak or a fish face or a mouse face. However, if you perturb that sonic hedgehog signaling, you get this very characteristically foreshortened face, which is really, really interesting. And a very similar pattern is found in FGF8 as well. So for those of you who are familiar with sonic hedgehog signaling, you might know that sonic hedgehog plays a massive role in development kind of all across the body, and is thought to be governed by a very vast, very complex supply jumping network. And because it has such a vast supply jumping network, it's thought that sonic hedgehog signaling and the network that controls it has a very large mutational target size. So if you break apart the nerve cranium into its 2 components, and you kind of visualize an underlying hypothetical pod jumping network, all we have to do to get a short face is perturb sonic hedgehog signaling. And if you're going to perturb sonic hedgehog signaling, you can hit the signaling network in so many different places to perturb that signaling that you can actually produce a superficially convergent phenotype, even though the actual mutations that are perturbing the signaling are not homologous, and the trajectories that are forming this face might not be similar. So that was kind of the mechanism that we hypothesized that was driving this convergence in brachycephalic skulls. There was this massive network. You could hit it in so many places to perturb the signaling that you were producing more brachycephalic phenotypes than you were elongated faces. And it got us thinking, is it actually easier to evolve a brachycephalic skull? So whenever you're trying to diagnose convergence via developmental bias, it's really tricky, because you actually need, as I just showed you need a lot of evidence in some very disparate fields of biology. So this was fun for me, because I had to kind of hop around and collaborate with a bunch of people and ask a bunch of very different questions. So the first thing we need to diagnose convergence via developmental bias is phylogenetic evidence of convergence. So we have that here with our nice tree. We know that there is indeed convergent evolution going on in skull shape for these electric fishes. The second thing you need is a biased developmental mechanism. So we have that sonic hedgehog signaling for the forebrain. The last thing that you need, which is the hardest, I would argue, is evidence of a wide range of adaptability, which is super vague, right? So because the developmental programming is producing these convergent phenotypes and not external environmental stimuli, it's thought that these convergent phenotypes are probably not really going to be tracking ecological stimuli in the first place. So now you have to test that. And so that brings us to almost our second question, which is, what are the adaptive implications of skull-shaped convergence in juveniformes, or these electric fishes? And so now we're completely on our second question, which is, how does the skull interact with its external environment? In order to answer this question, we have to actually quantify an ecological parameter associated with skull shape in these brachycephalic fishes. So I was really excited to do this part of the project because in undergrad, I had worked a lot with trophic ecology of billfishes. So I finally had a chance to study trophic ecology in these knife fishes. So one of the reasons why I picked trophic ecology as an ecological parameter for skull shape is because trophic ecology and diet has been implicated as a major driver of not only convergent skull shape evolution, but just skull shape evolution in general in vertebrates, right? The story of gnathostomes might be summed up as a story of diet, right? Like we evolved the jaw to go eat things. So diet plays a massive role in skull shape evolution. Um, However, I wasn't really satisfied with how diet had typically been quantified or described in a lot of studies, save for a couple cool studies by Nate Lujan and Ed Burris. For the most part, diet was described as discrete categories. This thing eats fish, this thing eats shells, and I wanted to continue. I wanted to figure out a way to quantify diet in a continuous manner. So I turned to stabilized slope analyses. Stabilized slope analyses are really cool because you can take like muscle clips or skin clips or scale clips, and you can figure out where an animal is feeding in its strut position. You can figure out what habitat it lives in, all kinds of really cool things. So for me, I was interested in two isotopes, really like 1.5. I was interested in the carbon-13 isotope and the N15 isotope. So carbon-13 can tell you things like carbon source pool and sometimes habitat, but the N15 isotope was what's super important because it actually enriches the trophic position. So if you want to figure out what a fish eats and you have its N15 values, typically the higher the N15 value, the higher the trophic position. So this gave us a really nice continuous way to track diet in these fishes. So the next thing we had to do was figure out a way to test the relationship between diet and skull shape. So I took a couple swings at this and a couple iterations, and this is the first iteration. So the first thing I wanted to do was pick a couple convergent regimes among brachycephalic skulls, and because of the nature of stabilized slope analysis, I wanted to pick things that lived in the same habitat. That way I didn't have to correct for different carbon source pools. So I picked two convergent regimes that can all be found in the Amazon River Channel. So we have our blue regime, which are some really, really rounded faces. And then we had the pink regime, which was more, more phylogenetically limited within our active nodes. And I asked a very, very kind of tight, specific question. So do specific trophic ecologies cluster within skull-shaped regime? Another way to look at this is if I grabbed the skull and all I knew was what regime it belonged to, could I then tell you what it ate? Very straightforward. So this was really fun because this question took me back to Peru again. We collected 58 species from these two regimes, and we took muscle plugs and we ran stabilized slope analysis to answer the question. Lastly, or after we had our stabilized slope analysis data, we ran a bunch of stats. I'm not really going to get into it, but the one thing I will say is that we used a linear discriminant analysis to test for differences in skull-shaped regime. So this was our initial iteration for diet data, and this is super, super exciting. So I transformed our isotope ratio to trophic position, and so now we have trophic position on our y-axis and the carbon-13 isotope on our x-axis. So I remember when I got these data, I was like at a gas station, and they had sent me the spreadsheet with all these data, and I hadn't done the transformation yet, but you could tell what the nitrogen isotope values, and I was like, man, this is really cool. They are eating different things. When I started this lab, an anonymous source told me that all these fishes were eating the same things. I won't tell you who that person was, but they told me that all these fish ate the same things. And so when I generated this plot, I was really excited because they're clearly not. So let me break it down for you. So remember I told you delta 13 carbon tells you carbon source pool. It tells you sometimes if these animals come from different habitats. In this case, we caught all these fishes in the same Amazonian River channel. So we don't expect them to differ along the x-axis. However, in trophic position, that's where all the action's happening. So we see this really nice stratification pattern with increasing trophic position. So there's clearly some separation in what these animals are eating. So then we looked at their guts to kind of ground truth what it was that we were exactly seeing. And we found that there were indeed at least 4 trophic gills within electric fishes. So we have this really cool piscivore gild where they're not eating whole parts of fishes. Electric fishes in this clade or in this regime These were specialized scale feeders. So they ate primarily the scales and tails of other electric fishes, and that was like the entirety of their diets. So they had these absurd choke positions. A lot of them were higher than some of the piranhas that we were catching them with, which is pretty nuts. The second guild we had were generalists. So generalists, you would find some fish tissue in there. Sometimes they would eat small armored catfishes, but they would also, you'd find aquatic arthropods in their guts as well. The next guild down these were invertebrates that ate primarily and almost exclusively aquatic arthropods. And the final guild here with Odontosynarchus being the only representative is the microinvertebrate guild. These animals are planktivores, so they spend their whole time up in the water column chasing plankton around. So when we ran our linear discriminant analysis, we wanted to see if scholarship regime could predict diet. And when you run these analyses, what you're looking for, if there are differences between groups, are nice non-overlapping kind of curves or humps. And we do not see that. So it's looking like scholarship regime does not really predict diet in this clade, which for me, at the time I was hunting developmental biases. So I was really excited about this. It's like, sweet, scholarship regime doesn't predict diet. So this was my last chapter of my dissertation. I wrote this up, sent it to another anonymous person, and they responded with this rebuttal, which basically they're saying, why are we surprised that the neurocranium, the upper jaw, isn't really tracking diet when its main job is to protect the brain? So why on earth? It's like, why are you looking for, pinky? Why are you looking for the ratio between pinky length and diet when that's not the function of the pinky? So basically saying the neurocranium protects the brain, doesn't really have a role in feeding. So this is not a surprising result. So I responded to this assault on my on my study with this really beautiful image. So here I have the skull of a electric fish, iodine stained to capture all the cranial muscles, and then CT scan. So here you can see where all the muscles are attaching across the skull. Here are the eyes, and here's the upper jaw. And I really want to draw your attention to the neurocranium kind of back here. So the neurocranium is nested in this kind of complex network of muscles that integrate the lower jaw and the rest of the body. So attaching to the dorsal roof of this neurocranium are a massive series of apaxial muscles that actually pull the head back during suction feeding. So it's super important. And then you have this adductor mandibulae muscle that powers jaw closing. So I show this image just to remind people that the neurocranium isn't some passive ride-along in the feeding process. Neurocranium is an active participant, right? The neurocranium is the base and is the center for all these activities. So all that's cool and all, but that didn't solve our problem, right? Like the neurocranium is not really a problem, but it didn't solve what we were looking for, where the neurocranium really wasn't predicting diet in any kind of sensible way. So if the neurocranium in the shape of the neurocranium doesn't predict diet, what does? So I just did a quick little analysis where I counted all the teeth on the lower jaw of these electric fishes, and I found that the number of teeth actually significantly predict diet. Where the more teeth you have, the higher your choke position. This is not surprising, right? Like at least within this clade, where fishes with lots of teeth tend to feed on things higher in throat position. But it got me thinking, and here I have, and I want you to kind of think about this too. Here I have the neurocraniums of these fishes colored by their convergent regime. And what you can see is, especially here in this blue regime, where the neurocraniums are actually quite a bit similar, but the lower jaw morphologies are widely divergent. So it's kind of crazy, right? It's like you have this very conserved shape up top, and down bottom, things are going kind of crazy. And the same can be said up here for the red regime as well. So I got really excited, and I wrote all this up, and I submitted it to a journal, and I'll tell you what happens next. But the major kind of point, the major point here that I took away from this, looking at all these really conserved neurocraniums and these wildly divergent jaw shapes, was that the neurocranium, especially the brachycephalic neurocranium, could really be compared to your 1998 Honda Civic, where you could adorn it and customize it with all these different things. You could put rims on it, change the paint job, put all kinds of crazy speakers in there, and it'll get you where you're going. It might not get you where you're going really, really fast, but it's at least widely adaptable. So you can do a ton of different things with it. And it might even get you, across the country once. So that was kind of the major takeaway. And again, I got really excited, wrote this up, submitted it, and I was swiftly defeated and rejected. The journal wasn't feeling it. They wanted us to kind of expand our sampling and use some more rigorous phylogenetic comparative methods. So this sent us back to the drawing board. It sent us back to kind of this central image where if skull shape alone doesn't predict diet, but aspects of jaw morphology do, maybe there's this pattern or process going on where different parts of the skull are responding to different selective regimes. So I decided to actually compare the rates of shape evolution between the neurocranium and the lower jaw. And remember, this is the first time that I've ever even thought about the lower jaw. So this is going to open up kind of a whole slew of new questions. So whenever you're talking about differential rates of evolution across an organism or across a particular organism, across different traits, what you're really talking about is mosaic evolution. So mosaic evolution and the Archaeopteryx, which is the poster child for mosaic evolution, refers to a pattern where different traits evolve at different rates. So the Archaeopteryx, when it was described as having the head of a reptile and the body of a bird. This is before people really realized that birds were in fact reptiles and not some gift from God. And as just brief aside, I always like to tell people and remind people that Archaeopteryx is really kind of the pinnacle for birds, right? Birds pretty much went downhill after this. So I don't know why they did it, but this was where they should have stopped. So again, when you're looking at the Archaeopteryx and you're thinking mosaic evolution, it could be said that the head of the Archaeopteryx was certainly evolving slower than the rest of its body. And that's why this kind of became the poster child for mosaic evolution. So patterns of mosaicism across an organism may be due to different levels of evolutionary constraint, but they can also be due to differing responses to selection. So those two things are really two sides of the same coin. If you're too constrained, then you're not going to be able to generate a large response to selection. So they're really kind of the same thing. One other cool thing about mosaic evolution is that it can facilitate morphological specialization, and we'll see kind of the implications of that downstream of this talk. And of course, mosaic evolution is, especially these days, is typically thought to be a product of evolutionary modularity. I introduced ontogenic modularity earlier, but now we're getting into the modularization of traits across deep time. So just a brief review, since I threw a lot at you, since I told you about integration. At the macroevolutionary timescale, integration among traits can have some very interesting consequences. And it's still, again, thought to be the result of pleiotropy or functional coupling. But now that you add now that you're adding in deep time, different things kind of come into play. So evolutionary integration, as it stands, can potentially constrain rates of trait evolution, because when trait complexes are too big, it becomes really, really difficult to find a mutation that's beneficial across all the different traits involved. So it's kind of like the Erykah Badu bag lady thing, where she has too many bags and can't catch her bus, right? There are too many traits, you can't distribute the mutation across all these traits because sometimes it's deleterious in some of the traits. So it becomes harder and harder to change and evolve. That being said, I would argue that evolutionary integration kind of gets a bad rap, because there are a lot of cases where evolutionary integration really comes in handy. So here we have a zebra. Let's consider the zebra. The zebra is a quadruped mammal. That means they walk on all four legs. And zebras typically have similar leg lengths all across the body. This is where evolutionary integration comes into play, because if you are locomoting on all four limbs, you definitely want all your limbs to be similar lengths. The last thing you want is the modularization of each individual limb, because you'll find that it's very, very hard to run if one of your legs is a surfboard, the other is a rocket launcher, and the other is a samurai sword. So this is where integration plays a very, very important role in tying together functionally relevant trait complexes. Evolutionary modularity is the rock star. Everybody really likes it. It's thought to facilitate morphological diversification and specialization, as I mentioned. So if we had the zebra in the first example, let's now consider the Kobe Bryant, rest in peace. So Kobe Bryant was a human. He was bipedal. So that means that he did all of his locomoting with his bottom limbs, which freed up his upper limbs, his arms, if you will, for a wide variety of tasks, including dunking on Yao Ming and beating Paul Pierce in the NBA Finals. So here we have two very, very kind of textbook cases of evolutionary modularity and evolutionary integration and some of those implications. So we've actually come a long way ever since the Archaeopteryx was described in quantifying mosaic evolution. So here we have this is a phenomenal study on birds, unfortunately, looking at rates of evolution across the bird skull. And this was a beautiful illustration of how mosaic evolution kind of works across the skull, where we have different aspects of the beak evolving more quickly than kind of the posterior portions of the skull, like the occiput and things like that. So this was to me one of the most brilliant illustrations of mosaic evolution to date. But fishes do it too. So fishes also, mosaic evolution has been found in fishes. Here we have a great study by Price et al. That found that functional traits in reef fishes evolve more quickly than non-reef fishes, and you can see that some traits are certainly evolving more quickly than others across these fishes. So this is another really good case of mosaic evolution. We also found it across body shape in fishes as well. I even took a crack at quantifying evolutionary integration in genodiforms. At the time, I only looked at the neurocranium again. So I looked at the degree of evolutionary covariation between the face and brain case and found that they were tightly covariated. There was high evolutionary integration, and as a result, they evolved at very similar rates. However, I did not examine the elements of the lower jaw. So now that we're back to the drawing board, now we're thinking about mosaic evolution, I kind of had to back up and think about a new way to attack this problem. So I decided to break this up into a couple questions. The first question was, are different regions of the skull, including the lower jaw now, integrated or modular in these electric fishes? Secondly, are there patterns of mosaic evolution across skull electric fishes? So are different regions of the electric fish skull evolving at different rates? Lastly, I was interested in, my last question was, what are the underlying patterns of mosaicism in electric fishes? And the clade I decided to answer this question in were the after-noted fishes. So this clade, I've showed you images of them kind of this whole talk, but I decided to focus primarily on this clade now. These are a deep channel specialized group of electric fishes. They have a lot of craniofacial disparity. In fact, the craniofacial disparity in Aptenotidae is equal to the disparity across the entire clade of electric fishes. So if you're interested in skull shape in electric fishes, this is a good clade to check. And now, instead of using two-dimensional geometric morphometrics, I had now made several trips at the Friday Harbor at this point and had amassed this very impressive CT scan data set. So now I was able to use three-dimensional geometric morphometrics to answer these questions. So for this project, I looked at 49 species aptenotids, which was a significant portion of their total clade diversity at the time. I included museum specimens, and I got to actually scan fishes that I caught in Peru myself. So this was really, really exciting. I used the same old phylogeny. I really just used the slide to show you my new skull tornado that now features fishes in 3D as opposed to 2D. But yeah, we use the same old phylogeny. The next thing that I was interested in was modeling the disparity through time of the different regions of the skull, and there was a reason for this. So if you're evolving in a non-constrained way, if you're evolving as quickly as possible under Brownian motion, then what you'd expect is that that your disparity should be proportional to your evolutionary rate. So with the disparity through time analysis, it gives you the really nice ability to fit your multivariate data to a Brownian motion model. So this is one way to test for whether or not different regions of the skull are constrained or limited in some way from evolving as quickly as they should be. So we fit different regions of the skull that we found to be modular to a Brownian motion model using a disparity through time analysis. So let's go into some of our results. So this is a phylomorphospace. This is kind of your textbook, initial visualization of shape diversity in any clade. So here we have our first principal axis of variation, which again corresponds to long and short faces. So it was nice to recover kind of the same pattern in 3D that we found in 2D. However, in PC2, what I learned was that the mandible was the second most important axis of shape variation in electric fishes. So a brief aside here. So in electric fishes, the neurocranium goes nuts. It does all kinds of crazy things. But when you get to other fishes like acanthamorphs and things, neurocranium becomes more conserved, and you see more action and more variance going on in the jaws. However, in this case, the mandible was still the second most important axis of shape variation. And for the first 4 years of my career, I had actually ignored it entirely. So that was fun. That was a fun realization. The next thing that we found was we found that a three module hypothesis instead of just the face and brain case was our highest supported hypothesis of modularity. We quantified modularity between those three regions using a couple different metrics, and it was giving us consistent answers. And when we decided to look at the rates of shape evolution across these different regions, we found some really, really cool things. So our three module hypothesis for modularity consisted of the face in yellow, the brain case in blue, and then the mandible. I have the sensorium here grayed out so it doesn't distract you. But so we had a three module hypothesis, and what we found was that when we looked at the rates of shape evolution, the mandible was flying, right? The mandible was evolving 4 times faster than either the face or brain case. And this makes sense. So if you think all the way back to before I got rejected from that journal, we had already known that the mandible was exhibiting more shape variation or more morphological variation than the nerve cranium. And now when we put shape coordinates on it, we're finding that very similar pattern. So when we looked at disparity for time, we found that the facial region was following a Brownian motion distribution. That means that the face was presumably evolving as quickly as it could, or as quickly as it could within reason. When we looked at the mandible was obviously following a Brownian motion distribution as the fastest evolving module. What was really interesting, though, was that the the brain case, the good old brain case, actually did not follow a Brownian motion distribution. So for the majority of its time across relative time, the brain case has exhibited a very kind of consistent shape. So it looks like the brain case is more constrained than either the face or the mandible. This is really interesting because the brain case and the face did not evolve at significantly different rates. And as you know, from a developmental standpoint, sonohedral signaling starts at the forebrain, so basically the brain case, and expands into the face. So even though the face is mathematically less constrained than the brain case, the face is tied to the brain case. It shares tissue linkages, so it can only go as fast as the brain case allows it to go. The mandible has no such problem, as I'll explain. So how are we getting these mosaic patterns of evolution? How is it that the mandible is evolving so much faster than the face of the brain case? And rather, what are driving these patterns of mosaicism across the skull? Is it development, like I kind of alluded to? Are there functional constraints, or is it trophic ecology? So let's take a look at development for a second. So the mandible exhibits kind of a semi-autonomy relative to the rest of the skull. It evolves from a completely different population of progenitor cells. It's evolved and it's kind of derived from the Meckel's cartilage, where I showed you that the sonic hedgehog signaling and a bunch of other genes play a massive role in the development of the upper jaw. So the mandible from Jump Street, kind of early in a fish's development, is doing something entirely different. And fishes can actually induce environmentally relevant changes in their mandible shape very early in development. This bone is highly plastic, so this bone is really kind of designed to closely track the environmental stimuli that it's experiencing. It's oftentimes one of the first bones to ossify too. So a fish will oftentimes in development ossify its kind of upper and lower jaws, its mandible and its premaxilla, while the rest of the skull kind of lags behind. And that is kind of a constant reminder of the importance of diet on these bones. But what about the brain case? So the brain case might be evolving more slowly because it's involved in a wide range of different functions, and all these different functions might kind of integrate it and constrain it because it's basically less free to evolve and respond to different selective stimuli. So juvenile forms are stereophytes in fishes, so that means that they have a Weberian apparatus that connects their inner ear, which is about here, their inner ear to their swim bladder. The brain case, of course, connects to the entire spinal column. I showed you earlier all the different muscle systems that articulate with the brain case. There are nerve tracts that enter in through the brain case. So the brain case is doing so many different things that there's a very good chance that it's simply not able to respond to all these different evolutionary stimuli very, very quickly, and that gives us a very constrained pattern of disparity through time. So you can kind of imagine the brain case as being on lockdown in terms of what it can do. So what about trophic ecology? So while I was scanning these electric fishes in Friday Harbor, I was amassing a larger dietary data set, if you will, looking at trophy position across a larger sample of these electric fishes. I'd also picked up some biomechanics training from my good friend Matt Coleman. And one of the things I was interested in was the mechanical advantage or the force transmission of the lower jaw and how it related to diet. So when we looked at the relationship between trophy position and diet, or trophy position and force transmission in the lower jaw, we found this really nice tight correlation where fishes with lower force transmissions typically, or lower mechanical advantages, typically fed higher in trophy position than fishes with lower mechanical advantages. So what does this mean? So fishes with lower force transmission, due to the force velocity trade-off, fishes with low force transmission are able to close their jaws more quickly. So if you're chasing fish, if you're trying to catch fish or catch invasive prey, it helps if you can close your jaw really quickly. And if you're feeding on things like aquatic arthropods or hard shell things, you're going to be able to generate a lot of force. And that's why one kind of one-off thing that pops up in a lot of these fishes with high force transmission, like Adenus monoparti, you see like really large teeth kind of further back here, further back along the jaw, because it's kind of closer to that hinge, and so they're able to actually generate more force along that jaw as they're biting down. So we found this really, really nice relationship between diet and aspects of jaw shape yet again. So it's looking like the mandible is evolving to closely track these kind of environmental stimuli, while the rest of the skull appear to be far more constrained. So to conclude, we found that the mandible exhibits a modular pattern of shape evolution and a mosaic pattern of shape evolution, where it's evolving far faster than the other bones in the skull. And we found that the actual performance of the mandible is tracking trophic ecology. And with that, I'll take any questions.
All right, thank you, Kory. If anybody has any questions, please type them in the chat. But to get us started, I just want to ask, so, you say you were really inspired by fish whenever you were a child, and your research could be applied to lots of different organismal systems. How did you actually become interested in becoming a scientist? So, you can be interested in fish, but what made you want to have that as a career?
Not gonna lie, I think growing up, when my parents did let me watch cartoons, I watched a lot of like Dexter's Lab and Jimmy Neutron, and I thought it was so cool that these guys, had these laboratories usually in their basement, and they were able to do all these really really cool experiments. And at the time, I was a kid, so I didn't know what an experiment was. I didn't, so I didn't have anything to say about. It. I always, wanted to work in the lab, so I always thought it was really really cool to do science in that way. So that kind of got me started, and then I think in high school, 10th grade, I took a biology class, and that biology class was really, really formative for me. And that got me kind of thinking like, oh man, these animals are actually really cool. I knew they were cool before, but I didn't know they were cool from like a studying perspective. I just thought they were cool to like watch on Animal Planet and stuff. So I think those two things kind of really got me going.
Awesome. So we have a question from Prasanta Chakrabarty. He says, great job. What's next for you? Also, it's interesting that the two convergent groups of weakly electric fishes, the neotropical electric knife fishes and the African mormyrids, also have short and long snouted species. Is feeding in similar habitats the only explanation for the repeated convergence?
Mm. That's interesting. So I guess I'll answer the second question. The second question first. So I don't know a ton about Mormyrids, unfortunately, but I do know that they do have highly piscivorous forms as well, like Mormyrops and things. So I think it might be. I think diet might be playing a massive role in that, along with developmental biases. But I don't know how often the short face horns have been produced in Mormyrids. So that question kind of remains really open in that way. Um, so to answer the first question, what's next? I just started this gig at Rice University, so that is next for me. We'll be continuing to set up this lab. I just got a CT scanner, so for everybody that's here, once the CT scanner is up and running at the end of November, early December, anybody who wants to scan something cool can certainly scan it here in Houston for free. So that's really what's next for me is mentoring grad students and getting this lab going.
Yeah, I guess kind of related to that. So you chose to take the academic route career-wise, and for those on the call who are graduate students trying to make the decision between, going into industry or going into academia, or the other various career paths you can take, what helped you make the decision that you wanted to go into academia? Like, what played into that?
So I did some undergraduate research, early undergraduate research at Nova. I didn't like really form my own project necessarily. I had like a small project that burned out really early. But one thing that I always thought was cool was that, I was like, I came from a place where in North Philadelphia where to get your money, you had to, actually work for it. There's like a massive blue collar workforce there. And you felt every dollar you earned. You felt it. And I remember sitting in these labs, I was like, man, these guys are getting paid to just sit here and think about what these fish eat. That is insane, right? That like blew my mind. So when I got to grad school, that was at the time, grad school was like the highest paying job I ever had. And so the idea that I could just sit there, I could have a good day, I could have a bad day, I should be getting paid to just sit there and think about these fish. I could write nothing on a piece of paper all day, and I will have done science because yeah, I would have at least considered fish. And right before grad school, I worked at a metal factory. And again, I felt every dollar that I made. So for me, research I thought was this really cool thing where you're able to kind of just, sit there alone with your mind or collaborate with people. That was another really fun thing where, you brought your own perspective to a project, to a series of questions, and then you linked up with other cool scientists who had completely different backgrounds, completely different expertise, and even, different walks of life. And we would, try to figure out what's going on in the system together. I just thought that was phenomenal.
Yeah. Okay, I'll take another audience question from John Friel. Any comments on the role of sexual selection on the head shapes in no, I don't know how to say it.
After
Aptenoid.
this from John. Yeah, so there's actually a whole like third of a talk here just looking at sexual selection in genetics, but sexual selection actually plays a massive role in skull shape evolution in genetiforms. So maybe I'll actually skip down to the bottom where I just show you their weird faces. So some genetiforms actually grow these really elongate faces to engage in intraspecific combat. So here's a Compsognathus amylae. They grow out these long pelican beaks to jaw fight, to like jaw lock and wrestle with each other. And this has actually these phenotypes have actually evolved multiple times independently. So yeah, that's what that's basically what a terminal male Compsognathus looks like in terms of jaw shape.
Cool. Okay, from Nick Mason, awesome talk. What about fish teeth? Do they vary among more myrid skulls in association with trophic ecology? What role might they have in intrinsic/extrinsic role in your broader framework of adaptive evolution and fish craniomorphology?
Oh God, teeth. So I honestly haven't really, ever since I counted teeth on the mandible, I haven't really looked at teeth. Teeth are like oftentimes their own like thing, but just kind of broadly and generally speaking, the piscivores in Aptenoideae typically had broad like conical teeth, as you would typically see in other piscivores. So I don't have much to say about teeth and diet, but genetiforms do another cool thing with their teeth in terms of sexual selection. So, yeah. So, yeah, this is a good one. So, this is a male Snarkagite nattereri, and so the males of these fishes during the breeding season will grow out these crazy teeth out of the front of their faces for jaw locking and fighting each other again, like I showed you in this video. What's crazy though is that the females of the species have no teeth whatsoever. So here's the mandible kind of segmented out away, and this is the female mandible where there are no teeth whatsoever. So these fishes were originally described as two. So when the male was found, it was actually put in its own genus altogether because it looks so different from from the females of the species. So I don't know very much about the teeth, how teeth track trophic ecology in this clade, but they certainly are tracking sexual selection.
So since we are part of the Office of Diversity, and diversity is something that we think about, what role do you think professors have in encouraging diversity and inclusion? And is that something you're thinking about when recruiting for your lab, since you're a new professor?
So professors have a really important role because professors are oftentimes the interface between upper administration and the undergraduate or graduate student body. And professors have a lot of power in a lot of cases, especially professors with labs, because they have the ability to literally kind of create, to basically bring in diversity and kind of create almost a pipeline for diverse grad student body to enter academia. So as I'm a professor, my goal and what I'm constantly trying to do is diversify academia. And for me, it's not like super hard where it's not like I have to go out of my way to like remember to do this because I am a Black scientist and I grew up around Black people. So for me, like creating a lab that has a bunch of people that look like me is not it's almost second nature. But I think it's really important to keep that in mind for other professors out there because increasing diversity does seriously increase the range of perspectives and the range of kind of walks of life that you can employ when you're trying to answer some of these questions.
Yeah, awesome. Did you have any role models growing up who were Black scientists that you saw, or no?
Uh, let's see. Not until I was already in grad school where I remember I ran into Scott Edwards in Austin in evolution. And I barely said a word to him. I remember I had like read through the list of some of the people who were kind of like a big part of the society. And I remember just seeing Scott Edwards, and it was so cool just seeing another Black man who studied evolution, even though he studies birds. I was really cool with that. I thought it was awesome. And I remember watching him walk through the hallways in Austin. I was like too shy to talk to him, but eventually he grabbed me and talked to me. So yeah, I guess later on, Scott Edwards was kind of that first role model for me.
Yeah. Does anyone else have any questions before we close out? Feel free to unmute yourself if you want. No? All right. Well, thank you so much again, Kory, for this awesome talk. And
No
thanks
problem.
everyone for coming.
Thanks for having me.